Hello, everyone. It's my great pleasure today to introduce Dr. Ashley Solmonson. Um, Ashley received her BS degree in biology and chemistry at Sam Houston State University. She then went on to receive her PhD in cell and molecular biology at the University of Texas in Austin. And after her PhD, she completed her postdoctoral training in Ralph's lab here at UT Southwestern. During her postdoctoral position, Ashley conducted foundational work showing that the placenta and the embryo use distinct strategies for energy production and biosynthesis. And in 2023, we were so excited to have Ashley join us as an assistant professor in the Green Center, where she is adding to our growing critical mass of card-carrying reproductive biologists. And to reflect that, Dr. Solmonson has a secondary appointment in OB-GYN here. Work in the Solmonson group focuses on mechanisms in which the placenta balances metabolite homeostasis across both the adult and the fetal compartments. Her group uses stable isotope labeling in pregnant mice, as well as a combination of mouse genetic models, and is informed by a deep understanding of human genetic, metabolomic, and pregnancy-related diseases. So with that, please join me in welcoming Dr. Solmonson.
Thank you so much, Ed. I'm short, so I'm going to push this down a bit. Yeah, thank you for the introduction and for the forum to talk to everybody about the work that we're doing in my lab. So my lab studies metabolism during development, and we know that metabolism is really important for development because it supports things like energy production and biomass production. But in more recent years, we've grown to appreciate that it also plays a role in epigenetic regulation and cell fate determination, and even developmental signaling and embryonic patterning. So my lab studies healthy developmental metabolism alongside two disease settings. The first is inborn errors of metabolism. So these are monogenic disorders caused by mutations in metabolic enzymes. And we think that these will teach us about the metabolic requirements for tissue development and formation. The other disease setting is pregnancy-associated diseases, primarily those that are derived from abnormal development of the placenta. This includes like preeclampsia and intrauterine growth restriction. And by studying these, we hope to understand what are the metabolic mechanisms of disease. So what ways abnormal metabolism contributes to the pathophysiology of disease. So we're primarily focused on the placenta. And so for those of you who are not placentologists, let me give you a little background on placental development. So the placenta is derived from fetal tissue, and specialized cells from the placenta called extravillous trophoblasts actually invade the maternal arteries in the uterus and remodel them. And they do so to make them high capacitance but low resistance. So you get increased perfusion into the tissue. And this allows for highly oxygenated and nutrient-rich blood to be exposed to syncytial trophoblasts, which are other specialized cell types in the placenta that facilitate nutrient transport and oxygen availability to fetal blood vessels. Now, if this doesn't happen properly, if it doesn't happen on the right time scale, or if it doesn't happen in the right way, you get abnormal spiral artery remodeling, and this leads to a depletion of oxygen and nutrients that are available to both the placenta and the fetus. And it also increases the production of reactive oxygen species, whether there is reduced oxygen or oxygen at the wrong time. So oxidative stress management is really essential for proper placental development. And we're interested in looking at that from a standpoint of metabolism and how are the normal systems of placental development equipped to help cells deal with this oxidative stress. So I won't give you too much metabolism for those of you who are not metabolically inclined, but we do know that the oxidative pentose phosphate pathway is a critical pathway that cells use to manage oxidative stress. So downstream of glucose catabolism, the cell can oxidize glucose and generate NADPH in the process. And NADPH is important for oxidizing glutathione disulfide, reducing glutathione disulfides, and it's also important for supporting lipid and steroid hormone biosynthesis that placental cells need to do to make estrogens and progesterones. So we have a hypothesis that the oxidative pentose phosphate pathway is critical for normal placental development, and it's involved in limiting oxidative tissue damage in the cells. So we started with a really simple experiment, and we just asked if we try to differentiate human trophoblast stem cells into syncytial trophoblasts, is there a requirement for specific nutrients? And so glucose and glutamine are both major nutrients used by cells to make energy and to manage oxidative stress. And so we first did an experiment where we induced differentiation of syncytial trophoblasts. And under normal conditions, can be shown by expression of the marker syndecan1. And if we do this in the absence of glucose, the cells cannot induce syndecan1 and don't seem to be differentiating. But if we leave out glutamine, they don't seem to care. We can also look at this by immunofluorescence. So these are the human trophoblast stem cells, and we can see that they're mononucleated and have normal organization. If we remove glucose from the media, they kind of get fat, but they don't differentiate because differentiation of syncytial trophoblasts is shown by fusion of these cells and they become mononucleated. And so that's what you see here. Even without glutamine in the media, they can still fuse and become mononucleated. And of course, if the media is consisting of both glucose and glutamine, they can still fuse and differentiate properly. So syncytial trophoblasts need glucose, but not glutamine to differentiate. But importantly, we were interested in this oxidative pentose phosphate pathway. So we asked if we inhibit one key enzyme in the oxidative pentose phosphate pathway, glucose-6-phosphate dehydrogenase, does this block differentiation as well? So in this experiment, we have either vehicle control or increasing concentrations of a glucose-6-phosphate dehydrogenase inhibitor, and we can see that the expression of syndecan1 is reduced in the highest levels of the inhibitor, suggesting that just inhibiting G6PD alone is enough to block differentiation of these cells. We can also look at this through immunofluorescence and cell morphology. So if we differentiate into syncytial trophoblasts, we get clear expression of syndecan1 as shown in green. But if we leave out glucose or if we treat with the inhibitor, we no longer get that expression. So now we're understanding what are the metabolic requirements to form the placenta, specifically syncytial trophoblasts. But why are we interested in G6PD, aside from the oxidative stress component? Well, one reason is that G6PD is one of the most common enzyme deficiencies in humans. There have been over 250 reported mutations in this gene. And oftentimes these are polymorphic mutations, meaning you have two different co-mutations that lead to a deficiency in the enzyme. And importantly, there is a residual amount of enzyme activity that is usually present in individuals with this deficiency that only when the cells need to produce more NADPH, like when they're stressed because of infection or because of a drug exposure, do you get some sort of clinical manifestation of this disorder. And so we're kind of wondering, is this a way in which pregnancy stress can actually manifest clinically in individuals who have mutations or variations in G6PD? So we have partnered with Dr. Chris Herrera, who is an excellent clinician in the OBGYN department. She has been doing a longitudinal study to look at women with chronic hypertension. So these women are at high risk for developing pregnancy complications like preeclampsia and growth restriction. And she's been using multiparametric imaging like MRI to look at perfusion of the placenta and development of the placenta overall. And then she's collecting plasma and placenta upon delivery. So we've taken that placental tissue and done metabolomics analysis. And you can see that there is a distinct profile shift in the individuals who have chronic hypertension versus the normal controls. Now, of our cohort, some of those people went on to get preeclampsia, whereas others did not. And we were interested in, is there a way in which the metabolism profile or the metabolic activity can tell us about the risk that some individuals have for developing preeclampsia? And so to ask this, we went into our placental metabolomics data. We looked at the metabolites involved in this pentose phosphate pathway, and we can see that there seems to be a trend towards an increasing level of these metabolites in the individuals that got preeclampsia, but not those who did not. Now, within that, there is a lot of variability here. So you have some people that have almost a tenfold increase in the level of these metabolites, and some people in the same group that have like a 50% level of the activity or the level of those metabolites. And so we're interested to see is the degree to which we see this elevation of these metabolites in the pentose phosphate pathway, is that linked to distinct variants that can be found in the G6PD gene in those populations? And in that way, is the sequence of the G6PD gene informative for us for risk stratification of individuals who are at high risk for those who will develop preeclampsia versus those who will not? So this work is ongoing, and we're trying to expand this cohort to a larger number of individuals. But in the meantime, we're limited in placental biology by model systems. So the placenta is probably the least studied organ of the body, and we don't really have a lot of tools necessary to really understand the molecular or even metabolic mechanisms that lead to disease of the placenta. So part of what we're doing to solve that problem is we're making organoids derived from human term placental tissue. And so we're kind of modeling this after what we've done, for decades in cancer biology, where we make cancer lines derived from tumors. We can do the same thing with the placenta, and we can make lines that are chronic hypertension lines, preeclampsia lines, gestational diabetes lines, et cetera, that have the genetic variability of our patient population, and hopefully can inform as to molecular or hopefully metabolic mechanisms that can influence disease progression. And so we plan to, we already have developed organoids from normal, healthy individuals, but we plan to do this with our chronic hypertension cohort as well. And we want to characterize the capacity for these organoids to develop and synthesize NADPH within the oxidative pentose phosphate pathway, but also through other pathways, and then correlate that to any genetic variants or pregnancy outcomes to hopefully improve the diagnostic tools that are available to the clinicians, but also then to test different therapeutic options. And I will leave you with this thought, which tells you that I am a Ralph trainee, that metabolic genetic variability can manifest under very different conditions. So often these manifest at birth. This is why we do the newborn screen. But at other times, these can manifest in toddlers or even in adults after things like infections or drug exposures. And so we would argue that pregnancy is a stressful condition and can induce metabolic conditions in people who are deficient and otherwise clinically asymptomatic. And we think that there's even a possibility that this metabolic genetic variability can influence the propensity for those individuals later on in life to develop either postpartum symptoms or even postnatal symptoms in the fetus that include increased risk for cardiovascular disease, glucose insulin homeostasis, and even obesity. So with that, I will thank the folks in my lab that are doing the work. Weiju Shu is my right-hand man in the lab and has been excellent. Andrea Woods is my own organoid whisperer. I gave her a protocol and she made it happen. And of course, Chris Herrera has been wonderful to collaborate with clinically. And of course, the patients who agree to help us with the research as well. So thank you.